Good day, guys. No, I mean Homo sapiens. Welcome back to the microbiology, Prescott's microbiology. This is the book written by, or full name is Prescott's Valley and Kynes. So, wait, give me a second. Okay, so this book we are reading it through since I thought this was a good idea since students usually don't read the books. So I'll be going through it the one by one. The first chapter is the history one, and we are currently on the path from where the history started, and we are currently in the middle of it, and we will be reaching by the end. I don't know how long it will take, but it's taking us a long way. So I can't say I am, a knowledgeable. Wait. I can't say I'm knowledgeable enough to give you all the explanation, but I'll explain the things which will come through if I know it, and if I don't, we are going to Google. And we are going to Google a lot in this life since, there are history, the world history, which I have no idea about. I am surprised at myself that I know more microbiology history than the other one, like Einstein's theory of relativity or things. So let's start without any further waiting. So we were at the Einstein theory of relativity, and we already discussed the rickets shows Rocky Mountain spotted fever. Wait, were we somewhere else? Am I? Wait. Did I already cover this? I feel like I have. No, we didn't talk about the World War I. I remember this. So anyway, rickettsia was the disease that caused the Rocky Mountain spotted fever, and now we are further telling it with the 1908 history, the first model T Ford. Okay, guys, what do you think the T Ford would mean? I didn't know that the T Ford was for the car. Let me search it. T Ford. Okay, guys, don't mind me if I am taking a little bit long, since last time my YouTube was stuck and the Google window wasn't working, so I am checking that with that as well. So let me see if it works fine this time. I don't want me searching all the way and you guys just listening and finding nothing on the screen. So T Ford, the Ford Model T is an automobile that was produced by the Ford Motor Company. Here, this thing. Now let's go to the images and see, this is the car, and here I thought that the Ford was the model by some kind of scientist. I think today is working fine. See, wait, let me do this and see if the net is working fine. Okay, I think it is working fine. So without any worries, we will go to further, and kindly text me if it is stuck and I'm talking and the Google window isn't moving. So after that, the 1911. So okay, by the time the Model T Ford was occurring, the rickettsia showed up and rickets discovered it, and then with that 1911 Rose discovered the virus that cause cancer. Why do I feel like I have already talked about this? Anyway, Rose was a scientist in 1911. He discovered a virus that caused cancer. I think cancer concept was there before that, but it was the first time that he discovered the virus, that the cause of the cancer was virus. Wait, let me search. What was the Rose discovered what virus? We as a microbiologist need to know that what virus cause cancer. Like all in the microbiology there is the names, is to learn the names. And if we don't even know the names, what else are we going to study? This here it is, the Rous-Sacroma virus. Guys, can you see it? Yeah, the Rous-Sacroma virus. It is the full name. And is there any description about it? It is by extracting the filterable agent from, okay, so he, from a chicken. Was it a chicken cancer? Oh my gosh, why did my mind currently directly went to the human cancer? Okay, so it's the chicken cancer, and he injected that into the healthy chicken and they developed the tumors. Okay. Sorry, I am kind of person like that just assume things on their own. So in 1914, after that, the World War I began. Guys, are you not interested in what World War I is? To be honest, I have no idea what the World War I was about. World War I. Let's see what it is about. World War I, also known as the Great War or the First World War, was a global conflict that took place. Okay, we know that it's in 1914 to 1918, but among which countries was that happening? So it's Russia, United States, Middle East, and then the Germany, Austria, Hungary, Turkey, and okay, so these were the countries, and they were fighting against the France, Great Britain, Russia, Italy, Japan, and then again the United States. Okay, we are not going to understand it by reading the words, so let's just, okay, on map, see which countries were together against what countries. On map, let's go. So as we can see here, I think this red one is this one. See, we learned, the name Hungary or things. I think these red indicates, okay, this one is a good one. Let's see the red ones, the United Kingdom, the France, the Italy, the Greece. What others are this? Russia, Romania. So they were fighting against the Germany, Austria. Okay, but it doesn't contain the whole world. Like, look at this black picture. Like, it, where is the, see, if this is Asia, this is Africa, and this is America, this is Europe. So the whole thing isn't involved, almost not involved. Anyway, so the World War I was happening, and I don't know for what reason that was happening, but whatever, we are already passed through it. We should be worrying about the World War IV or something. In 1914 to 1917, the D. Hurleys and the Todd discovered the bacterial viruses. Okay, so I'll just tell you this. The bacterial viruses are called as bacteriophages as well, since these are the viruses that infect the bacteria. So they don't infect the humans. So I didn't know it was that in the past, that 1915 and 1917, that they were discovered, the bacteriophages. I think he would be culturing the bacteria and looked like the bacteria was infected with some kind of things, and then he studied that these were the viruses that actually infected his bacteria. And then what? This was the 1915 to 1917. And then what was happening in the world? Russian Revolution. Oh my God, Russian Revolution. After the World War I? No, World War I was till 1918, and this is the Russian Revolution. Wait, let me go back and see Russian Revolution. What happened to Russia now? It's either America, Russia, Germany, or few countries here and there that's always involved in things like that. Was a period of political and social change, began in 1917 and continued till 1923. What was the purpose of this? What was the purpose of it? Let's read the summaries. Two revolutions. The first, which in what actually was happening? What was happening? What was happening? So the Russian Revolution 1917 was a period of widespread dissatisfaction and economic relation that led to overthrow the imperial government and the establishment of Bolshevik rule. Okay, I kind of get it. So it was because of the people. So anyway, we know now that, well, there was Russian Revolution happening and there was World War I happening, and there the scientists were stuck with these microorganisms. One was discovering the bacteriophages or bacterial viruses, and the Fleming was discovering the lysozymes. We all know who Fleming is. Alexander Fleming is the same scientist. Now the 1923, the first edition of the Burgess Manual. Guys, I'll tell you that this Burgess Manual is so important in microbiology, you won't believe it. Without this, you can't even identify the microbes like completely. I can't even, I don't know, Burgess Manual is so long, and I can't even show you how in Burgess Manual there are the classification, the tests. The biochemical tests are there, and it is for every kind of bacteria it is different. So if you want to find some bacteria, you have to go through all these biochemical tests, and then in the end you are going to say that that these reactions follows the Burgess Manual, and this is this bacteria. It doesn't mean that it will do all the reaction. It just shows that it will do these reaction and it won't do these reactions. So that's that bacteria. So the Burgess Manual is so important. See, here it is. Like for example this one. Let me enlarge it a little bit. Okay, I didn't mean to click it. Go away. Why isn't it? When my computer isn't listening to me, this makes me irritated. Okay, see this here, this characteristics, and this is Klebsiella pneumoniae. It is just one bacteria, and it has these much of biochemistry. I mean, these are the tests, and these are the amino acids it can use or it cannot use. Like for example, oxidase activities, urea hydrolyzed, pectate hydrolyzed, OPNG test. And for example, all of these, we will be covering these in the later part of the book. Utilization of D-alanine, erythritol or galacturonate, and the rhamnose, etc. Like these are the amino acids and these are the tests. And it doesn't have to follow all of these tests. Like I said, it can go negative way or positive way, but it'll just show that what tests are required to isolate or identify the kind of microorganisms we are studying. So in 1923, oh my gosh, it's such a long history. 1923, and this is 2025. I think 1 century passed. Now, 1 century, or what do you call 1 decade? No, century. Century passed. Now 1927, Lindbergh's transatlantic flight. Guys, at first I thought this was Antarctica, right? The cold part of the world, the earth, but this is just the ocean. And Lindbergh is a person that I think made the, aeroplane, ancient type of aeroplane. First were 2 brothers, Wright brothers, They first discovered the powered aircraft, and sooner in 1903 were they discovering that, and in 1927 the Lindberghs already tested the flight, and it was no child's flight. It was above the Atlantic Ocean. Let me show you in the Google. What are they talking about? Transatlantic flight by who? Oh my god, I forgot. Lindberghs. Guys, my screens are separate, so I cannot see the writing. Just like you'll say, oh, look above, and you'll find, why is my writing wrong or spelling wrong, whatever, they will understand. So this is, I think, the Lindbergh, the scientist, and this one shows that the arrows, that this is the Atlantic Ocean, and look at the star. It started from New York and landed on somewhere else. Or wait, why not we study it correctly? Let's go here and they will tell us. So okay, St. Louis, the spirit of St. Louis. Oh my god, this is the name. So the departure was in Garden City from Roosevelt Field. Okay, he took off from this field in the Garden City and he arrived at the Paris in France in 33 hours later. Just think about these days. Nowadays we just have to take flight. I don't think it will take 33 hours, more just take maybe 1 hour. Guys who are familiar with the maps or the flight things, you can comment how long will it take for us to fly nowadays. Anyway, so by the time this was happening, the Griffith discovered the bacterial transformation. Okay, bacterial transformation is the process where the bacteria take up the DNA from the environment and wait, let me close this. Take up the DNA from the environment and integrate it in its own DNA, just so it upgrades itself. The one, and it can happen that they put the DNA in the wrong path in their own DNA, or the DNA they took from the environment isn't the good one, so they die. But the ones that survive are always the fittest version of themselves. So it is a phenomena, and we call it the sexual reproduction of the bacteria, and it is the vertical, what do you call it, vertical? One is horizontal transfer, which happens from one generation to another, like from parent cell to daughter cell, while in bacterial sexual reproduction it is the vertical transfer. Bacterial transformation, I'll show you just in the pictures. You can see here it was written. See, it is a transformation process that allows the bacteria to take up the foreign DNA, which can give them the new genetic traits. See, and now the image I'll just show you. I think it's not that, but it is better to look at the pictures, so understanding is better. Is there any clear picture? Any clear one? See here, this one. My mouse, you see my hand, small hand. This is the plasmid DNA. Bacteria usually have the plasmid DNA or the chromosomal DNA, and environment, I think the plasmid one survives more than the other one. So the plasmid DNA is in the environment, and this is the bacterial cells. It doesn't have any other plasmid, this DNA, the circular DNA, and it takes it from the environment, and when it integrates itself, so it becomes the transformed cell, and it is the upgraded version. And beside, you see, this is conjugation. You don't have to worry about this. We haven't discovered it yet. We have just discovered the transformation for now, and the conjugation is another sexual reproduction by bacteria. So let's move further. Griffith discovered, guys, this will be coming in your MCQs, or this is important. People ask that since it's so basic. Bacterial transformation. Now the 1929 stock market crash. Okay, this market is crash is not talking about the whole world's market. I think this is just talking about the America, I think. Wait, I don't wanna be hated for giving wrong information, but we'll search it. 1929 market crash, stock market. Yeah, let's see which country was it that the market was crashing for. Also known as Great Crash, was the major stock market crash in the United States. See, this is the United States. Okay, it's not the United States of America. Yes, it is. It is the United States of America. So the United States had the stock market crash. So At that time, 1929, Fleming said, let's forget about the market crash. We are more the penicillin is more important. So penicillin is the first antibiotic. I think all the microbiologists will know that. Penicillin is produced by the Penicillium notatum. It is the fungi name that produced the penicillin, and the Fleming was actually experimenting with fungi, the Penicillium notatum, or was it Spirogenum? No, so I think it was Spirogenum that the Fleming was experimenting on. And when he saw another day, no, wait, what was the story about? But it was by accident. It was by accident that the Fleming discovered the penicillin. Man, I have to go through everything. My mind is jumbled. Fleming, I can't even write the spelling. Fleming, discovery of penicillin. I hope they tell me the whole story. Okay, yeah. So he was actually growing the bacteria. He was the step. Okay. Sorry, I was a bit wrong about it. Not a bit, a whole lot. But it wasn't a fungi that it was growing. It was a Staphylococcus bacteria that he was growing on the petri dish. But the other day when he woke up, the bacteria wasn't growing a certain area, and it was because there was the penicillium fungi producing the penicillin, the antibiotic. So that's how he discovered the penicillin. So this was the story. So now we are going to the further discovery. It was when Nile studied for synthetic bacteria. Guys, for the synthetic bacteria are our saviors. And it is this late in 1931 that when Nile studying these bacteria, we should have done that sooner. You guys know cyanobacteria produce the oxygen that we are having in the atmosphere, or that were needed for the starting survival of the microorganisms. Microorganisms are the macroorganisms. So in 1931 when Nile studied the photosynthetic bacteria, so this is just the starting of photosynthetic bacteria. Now the 1933, the Prescott develops an electron microscope. Okay, it is this late that we are actually developing an electron microscope. If we go back and back, let's go farther back. It's even very back in my book as well. Not my book, all of you guys' books, but here you go. Wait, and I'm not a robot, so I don't know what date was that. So it's 1590 to 1608 that the Janssen developed the first compound microscope. 1590. Can you guys believe this? How many centuries later were we developing the electron microscope? But actually electron microscope isn't that easy to be made. But after studying the electron microscope over and over again, its structure, it became easy. So let me just tell you what electron microscope structure looks like. I hope they have given the image and I don't have to search for more. So yeah, here, look at the mid one. While the real one looked like the left one, while this one, the inner part looks like this, the middle area. So it is very simple in its construction, but I think people needed to know the chemistry behind everything. That's why it took so long to discover, or people didn't even know that the electrons could help us see the microorganisms. It's actually a very big discovery. So let's look at this. Oh my, why did I have to click? Why did I have to? Now I have ruined everything. Wait, okay. Oh my god. Anyway, you guys can see this area. This is the electron gun. This is made up of some kind of metal, and it releases the electrons when we provide it electricity. And then those electrons are actually very concentrated, and if they are not concentrated, they are made concentrated by this anode. that is like see in the ring form, and they go through further layers, so they are aligned and they directly fall on the microorganisms, or they are actually helpful in studying the cells as well. They put the whole, what do you call this, a mosquito or insect on the electron microscope, but we don't usually do this. We don't usually put the whole insect on the microscope. We made the smears on the slides, and then we studied it under the electron microscope, since electrons are very small and its penetration ability is very high, but not that high that we can use the whole insect on it. So further we are going. Now the electron microscope was done. Then there comes the Hitler. Guys, I think even a person like me have heard about the name Hitler, and he became the chancellor of Germany. Guys, he killed some people. I remember this. Yeah, he killed the Jewish people. Yeah, is it a taboo or something? But he killed so many people, and then he died, but he didn't die by the natural death. He died by suicide. Why will a guy like that killed those many people do the suicide? And let's see. Wait. Okay, I thought my live ended, like I told you. Wait a second, is my live working okay? Like I told you before, guys, I don't want this to go the wrong way, like as usual. Okay, yes, thank goodness it's working. I didn't want to go wrong. So why Hitler suicide? Did the suicide or did the suicide? Whatever, they will know. So all, and then the Hitler and the dictator of Nazi Germany, gunshot to head. What? Yeah, suicide via gunshot to head. But why did he do it? Yeah, I remember it now. They say that his troops entered the heart of Berlin and Hitler committed suicide because he thought that the victory wasn't going to come to him, and he just did the suicide. I think after the victory went to the other side. Wait, who was he fighting with again? He was especially, yeah, Russians. See, he knew if he lost against Russia, he wasn't going to have it peaceful. So he thought, why not die at my own hands than giving myself to other people? Must be his own thought. Otherwise, why would he do the suicide after becoming the Chancellor of Germany anyway in 1933? Now, in 1937, Krebs discovered the citric acid cycle. The Krebs that has given us headaches in the papers of 12th biology or in the 10th biology. Citric acid cycle, the Krebs cycle or the citric acid cycle, and it was discovered by 1937. I don't think I have to go dive into this one, since we all know that it is the middle phase after the glycolysis and before the electron transport chain that we actually go through to produce energy or electrons. So then the 1939, it actually produced a lot of NADH or FADH2, and that goes to the electron transport chain to produce the ATPs. Then the 1939 World War II begins. World War II. We already have seen that the World War I was actually among the larger countries, but I think World War II is going to, World War I was among the large countries. World War II, I think, is the, going to be involving smaller countries as well. So let's just directly go and see it on map, who are fighting against who. Wait, it isn't showing it. So where is it? There should be a clear diagram in there. Is it this and that? No. Is this one correct? These ones? Or this one? Yeah, I think it's this one. See the blue areas and the purple area? Yeah. So Russia, America, Africa. Is this Africa? Is this Africa? Okay, I'm not going to take names so I can save myself from embarrassment later on. So who won this? I think it's obvious that these countries have larger area compared to these one. Who won this? Wait. So who won? Who won? The nationalists won the civil war and favored the Axis. Wait. Germany quickly overran much of the Europe and was victorious. Okay. Guys, this area is Europe, right? Isn't this Europe, this area? So it looks like the area which was dominant even in the structure 1. Let's see it again. Seems like the Axis and the exiles are the allies. Sorry, Axis and the allies are the real categories. So why not look at that? Wait. Where are we going? Here, this picture. The Axis and the allies. See the blue area hold the allies. I think white. This area is white. Is this not involved in the war? Looks like the white area isn't involved in the war, and the Axis one are smaller, and this area comes in the Europe. I think this area. While the diagrams doesn't looks like, Wait. Let me first see where the Europe is. Okay, so this area. Yeah, it was shown in the images that it was at the side. See here, this area, this red area, this is the Europe. So they are saying that the Germany won war against the Europe. And what about the other countries? Were they not? Okay, we are not diving deep. So World War II, you guys can watch any other video on World War II for your information. I'm not going to watch it. I have no business watching that. 1941, Beadle and Tatum proposed one gene, one enzyme theory. Okay. We are not going to Google for this one. I'm just going to tell you that Beadle and Tatum were the scientists, and they proposed the theory that one gene was enough for one enzyme production. They said that one enzyme was produced by only one gene, like one gene was sufficient for production of one enzyme. You guys get it? But these days, no, it's possible. No, it's actually very naive to think that. Okay. So these days we know that there are multiple genes involved in the production of one kind of particle like enzyme. Enzyme is very complex and have multiple factors, cofactor area or the chains, the polypeptide chains, etc. So yes, there are not just one gene, there can be multiple genes involved in the production of enzymes. So okay, gosh, we are going to the good story. It is the 1945 atomic bomb dropped on the Hiroshima. Poor guys. But I think we are already seeing that okay, this was the America that dropped bomb on the Hiroshima. So I searched about it. If I am wrong, correct me. I have no sites or not siding with any country. I don't know. Okay, will this tell you guys that I'm not from America or Japan? So anyway, they dropped a bomb. Okay, I'm finishing on this page since I have some work to do. I just wanted to do live since I haven't done it in few days, and by the pattern I am going live, this will take a whole lot for us to cover this book, complete this book. So don't worry, there will be days when I will be going live a lot longer, so we can complete this book. And I think my pace is slow as well. Don't worry, as soon as we come out of this history, our pace will be a bit better, since this side of history I'm not very familiar with it, and I can just go pass through it without studying it. So good luck to you guys in life. Do you have Homo sapiens? And to me as well. I'll be returning for the further study. Anyway, bye. This is the